recently awarded federal approval to test out a lab-developed corn variant that is believed to be resistant to arid environments and droughts. All right, we deserve to know what we are eating, added Van Gosen. Virtually every major country requires labeling of GMOs in foods so their citizens can make informed choices, including all of Europe, Japan, and even China. Monsanto's laboring dollars are pouring into politicians, so it's clear we are a GMO contaminated U.S. Congress that threatens our health and the health of the plant. Occupy Monsanto rallied outside the Cannon building in Washington's Capitol Hill on Friday, where hazmat suits clad GCU officers identified victims of genetic crimes. Other protests were planned around the world to be held at the same time, including demonstrations in Los Angeles, Seattle, whatever that word is, in Winnipeg. In September, Occupy Monsanto is planning an even larger global protest that is said will become a day the biotech firm will never forget. Alright. And there you got devils. That's a devil right there. Bunch of butchie. Look at these people, man. They don't even. Look at that. You know it's set up. Then you got that simple ass nigga right there. Okay. Dumb, stupid ass jakes, man. Alright. So they getting shine time, but that's all set up. Okay. Alright. So let me get a few more scriptures. Hold on, let me touch this. Something about this stuff. Alright. Alright. Production. GMOs. Genetically modified involves the insertion or deletion of genes. When genes are inserted, they usually come from a different species, which is a form of horizontal gene transfer. In nature, this can occur when exogenous DNA penetrates the cell membrane for any reason. To do this artificially may require attaching the gene to a virus or just physically inserting the extra DNA into the nucleus of the intended host with a very small sirdine or with very small particles fired from a gene gun. However, other methods explore natural forms of genotransfer, such as the ability of argobacterialism to transfer genetically material to plants, or the ability of lentivirus to transfer genes to animal cells. The history. The general principle of producing a GMO is to add new genetic material into an organism's genome. This is called genetic engineering and was made possible through the discovery of DNA and the creation of the first recombinant bacteria in 1973. An extensive bacterial E. coli expressing as the exogene Salmonella gene. This led to concerns in a scientific community about potential risks from genetic engineering, which were first discussed in depth at the Issachar conference in 1975. One of the main recommendations from this meeting was that government oversight of recombinating DNA research should be established until the technology was deemed safe. Okay, I'm gonna skip down. So they doing this, all right, and they've been doing this from the 1975. So, man, so they already know, all right. They supposed to have been waiting to see if it was safe. They've been testing it without even really checking to see if it's safe or not. So, a lot of these foods can cause a lot of health problems and destroy people. Okay, it says small-scale experimental plantings of genetically modified plants began in Canada and the U.S. in the late 80s. The first approvals for large-scale commercial cultivation came in the mid-90s. Since that time, adoption of GM plants by farmers has increased annually. Why? Because that food, those seeds destroy other seeds. Okay. They even doing it on animals and everything, man. Okay, skip down to this plant. Transgenic plants. Transgenic plants have been engineered to possess several disturbing traits, such as resistance to pests, herbicide, or harsh environmental conditions, improve product shelf life, and increase nutrient value. Nutrient value. Since the first commercial cultivation of genetic modified plants in 1996, they have been modified to be tolerant to the herbicide glufosinate and glufosinate, to be resistant to virus damage as a rain spot virus resistant GMO papaya grown in Hawaii, to the production of Botox, an insert that is alleged non-toxin to mammals. Okay, most GM crops grow today has been modified with input traits, which provides benefit mainly to farmers. The GMO oilseed crop on the market today offer improved oil profiles for proceeding or healthier and by oils. The GMO crop is developed often offer a wide range of environmental and consumer benefits, such as nutritional enhancement and drought and stress tolerance. GM plants have been developed by both private corporate companies and public research institutions such as the CIMMYT, the International Maize and Wheat Improvement Central. Other examples included genetic modified sweet potato enhanced with proteins and other nutrients, white golden rice developed by the International Rice Research Institution, the IRRI, has been discussed as a possible cure for vitamin A deficiency. Scientists of the University of York developed a weed that contains genes from bacteria that can clean up TNT and RDX exposure contaminants from the soil. It was hoped that this weed would eliminate this pollution. 16 million perpetrates service. Okay. Yeah, so they trying to create plants. They make it sound all good, but let me go get to the part we talk about health problems. The Terminator or Traitor. Often cite controversy is the technological protection of the Terminator. This uncommercialized technology will allow the production of first generation crops that will not generate seeds in the second generation because the plant yield sterile seeds. The planting for this so-called Terminator gene technology is often only by Data and Panland Company and the United States Department of Agriculture. Delta and Pineland was bought by Monsanto Company in August 2006. Similarly, the high proteinable trait specific genetic use resistant technology, also known as the Traitor or T-DRENT, requires application of a chemical to genetically modify crops to react engineer traits. This technology is intended both to limit the spread of genetically engineered plants and to require farmers to pay yearly to reactivate the genetic engineered traits of their crops. Genetically used retraition technologies under development by companies including Monsanto and AstraZeneca. In addition to the commercial protection of technology is self-pollinating crop such as soybean, a genetically constructed issue. Another possible of the terminated gene is to prevent the escape of genetically modified traits from protruding crops into wild type species by sterilizing any resulting hybrids. Some environmentalist groups, while considering outcrossing GM plant dangers, feel the technology will prevent reuse of seed by farmers growing such Terminator variants in the developing world, and is ostensibly a means to exercise plantal claims. However, other environmental groups welcome the Terminator gene as a means of preventing GMO crops from mixing with natural crops. Hybrid seeds were commonly used in developing countries long before the introduction of GMO crops. Some hybrid crop seeds cannot be saved, so purchasing new seeds every year is already a standard agriculture practice for a majority of farmers. There are technologies involving that contains the transgene by biologically means and still can provide fertile seeds using fertility restore functions. Such methods are being developed by several EU research programs. Among the contenders of co-excerpt. Alright, that's just to back up what I said before about them having those seeds that destroy other seeds, or that you won't be able to make you won't be able to have seeds that grow naturally. So this man is deaf, man. Let me get this real quick. John 10:10. It says, The thief cometh not but for to steal and to kill and to destroy. Alright? And that thief is talking about the so-called white man. That's his MO. To steal, to kill, and